Hello, everyone, and welcome to part 4 of our webinar series, Irrigation of Controlled Environment Crops, Balancing Light, Water, and Nutrients. Today's presentation will be about 30 minutes, followed by about 10 minutes of Q&A with our presenter, Dr. Gaylon Campbell, whom I'll introduce in just a moment. But before we start, we've got a couple of housekeeping items. First, we want this webinar to be interactive, so we encourage you to submit any and all questions in the questions pane, and we'll be keeping track of these for the Q&A session toward the end. Second, if you want us to go back or repeat something you missed, don't worry. We will be sending around a recording of the webinar via email within the next 3 to 5 business days. And all right, with all of that out of the way, let's get started. Today, we'll hear from Dr. Gaylon Campbell, who will discuss how to get the most efficiency and production out of your greenhouse crop. Dr. Campbell has been a research scientist and engineer at Meter for over 20 years, following nearly 30 years on faculty at Washington State University. His first experience with environmental measurement came in the lab of Sterling Taylor at Utah State University, making water potential measurements to understand plant water status. Dr. Campbell is one of the world's foremost authorities on physical measurements in the soil-plant-atmosphere continuum. His book, written with Dr. John Norman on environmental biophysics, provides a critical foundation for anyone interested in understanding the physics of the natural world. He has written 3 books, over 100 refereed journal articles and book chapters, and has several patents. So without further ado, I'll hand it over to Gaylon to get us started.
Well, thank you. And thanks for being with us today to talk about balancing light, water, and nutrients in controlled environment crops. Now, when you start the production cycle, you start with a mostly empty room like the one that you see here. 8 or 9 weeks later, the room is mostly full with biomass, like you see in this picture. Well, where did all that biomass come from? Typical amount might be 4 kilograms per square meter. Now, we supplied a lot of water through irrigation for the plants. We assume the vegetation is around 70% water. That'd be around 2.8 kilograms. Now that leaves 1.2 kilograms per square meter of dry matter. Now we put some fertilizer in the water, and the plants consumed much of that. The dry matter is around 10% nutrients, and so we could subtract that too. And we come out with something like 1.1 kilograms per square meter, still at that point. Now, to go into this in a little more depth, let's return to this picture of a plant that we used in an earlier seminar. It showed the 9 environmental variables or parameters that represent the main connections between the plant and its environment. We said it's useful to think of some of these environmental parameters, such as light, carbon dioxide, water, and nutrients, as resources. And consider the factors that enable the plant to capture those resources. Assimilation is the process by which carbon dioxide, water, and light from the plant environment are captured and converted to carbohydrate. To grow, the plant captures nutrients and water, combines those with the carbohydrate produced by assimilation, and makes those into stems, leaves, roots, flowers, and fruits that make up the biomass of the plant. We also noted that the third set of processes listed here occurs simultaneously with the other 2, but doesn't capture resources. Development is the progression of a plant through recognizable phases. The rate of development can be influenced by availability of resources, but it's most strongly influenced by temperature and day length. On the other hand, development can strongly influence the plant's resource capture, since it controls the time in each of the phases. In controlled environment crop production, all the variables listed here are under the control of the grower. It's the job of the grower to choose the values of each of those variables to attain the goals of the production facility. To properly control those variables, the grower needs to know what values they have, and how they influence the outcome of the production process. The Arroyo production platform is capable of monitoring those variables and reporting their values to the grower. So the 1.1 kilogram per square meter comes from the light and the carbon dioxide, kind of like magic, converting light and air to biomass. Let's look in more detail at how that happens. But first, I want to consider the idea of limiting resources in crop production. About 150 years ago, Justus von Liebig gave us Liebig's law, and it states that growth is dictated not by total resource availability, but by the scarcest resource. It's often illustrated using Liebig's barrel. Here the staves represent the various resources. Mineral nutrients are typically the resources that are illustrated by the barrel, but I've added the environmental ones that we just talked about in the previous slide. In nature, any of these resources could be limiting, but in controlled environments, all of them can be optimized to get maximum production or maximum yield with minimal waste. In many production facilities, the measurements would be too difficult to get a clear picture of what the limitations are. But in controlled environments, the Arroyo production platform provides all the information that we need. Now, a lot is going on in the plant to take it from the small size you saw in the first slide to the full room that you saw in the second. We can lump some of those things into three main sets of processes, and we can model those processes. Studying these models in detail will give us insight into how we can balance resources for maximum production. The three processes that we'll consider are the conversion of photons to biomass, the conversion of water to biomass, and the conversion of nutrients to biomass. This is a light response curve for a leaf. The rate of uptake of carbon dioxide by the leaf is plotted here as a function of the photosynthetic photon flux density, or the light level on the leaf. At 0 ppfd, the assimilation rate is negative. So the leaf is not carrying on photosynthesis. It's turning carbohydrate that it had into energy that it can use for metabolic processes. As the light increases, the assimilation increases. At these low light levels, each molecule of CO2 takes about 12 photons to convert it to carbohydrate. The theoretical minimum is 9, but in real leaves there are some inefficiencies. The horizontal axis goes to 2,000 micromoles per square meter per second, and that's around the ppfd of full sun. Some indoor facilities approach that value, but most are quite a bit lower than that, maybe around half in a good facility. Note that the higher the light level, the lower the slope. Therefore, the less the efficiency the light use. At these high light levels, something else is becoming the limiting resource. Now, if we increase the carbon dioxide concentration, the assimilation rate also increases. The blue curve here shows the response from the previous slide, and that's for ambient CO2 around 400 parts per million. The gray curve is for 1,000 parts per million. We think about Liebig's barrel, doesn't do much good to supply more light if CO2 is limiting our yield. To get the good out of the high light levels, you need high CO2 levels too. Now, a useful way to think about the amount of light available for photosynthesis is the daily light integral, or DLI. In outdoor environments where light's always changing, computing this number can be a challenge, but in a growth room, it's easy. If you had a growth room with a light density of 1,000 micromoles per square meter per second, and the light were on for 12 hours per day, the daily light integral would be 43.2 moles per day, as shown in the calculation here. Now, if you want to know how much biomass we could produce with that many photons, we'd multiply the daily light integral by a light use efficiency and the total time the crop was growing. The light use efficiencies vary depending on CO2 levels and other variables, but 1 gram per mole can be thought of as a kind of an upper limit. If we multiply these together, we get 2.6 kilograms per square meter of dry matter produced. We can compare that with the 1.1 kilogram per square meter value that we talked about a little earlier. Now, why might this one be lower? I've listed several possible reasons here. First, part of the energy we fix in photosynthesis goes to carrying on metabolic processes within the plant. We call that respiration. These processes are temperature dependent, so more assimilate is lost to respiration at high temperatures than at low. It's therefore good to decrease nighttime temperatures to reduce respiration losses. The next factor is fractional light interception. The canopy can't capture 100% of the incident radiation, and photons that aren't captured by leaves can't produce dry matter. And finally, we saw in the light response curves that higher light levels fix less CO2 per mole of light than low light levels do. Now let's consider our second model, how we convert water to biomass. Let's start by looking at the table in the right side of the slide. Water participates in biomass production in several ways. The photosynthesis equation we saw a few slides back showed water, CO2, and light combining to form carbohydrate. It takes 0.6 grams of water for each gram of dry matter that we get. We also talked about something like 70% of the plant structure being water, and this takes an additional 2.4 grams of water per gram of dry matter. But the transpiration process, shown in the diagram on the left, so thoroughly outdoes the other two processes that we don't even worry about, the water used in the first two. Consider that diagram. Now, the carbon the plant fixes must come from the air, but the metabolic processes that fix the CO2 must go on in a highly hydrated environment. We talked about that in the first seminar, showing that inside the leaf, the relative humidity is very near 100%, while the humidity outside the leaf may be half that. The diagram that I show here is a cross-section of a plant leaf. We see a pore or a stomate in the leaf through which the CO2 diffuses to be fixed by photosynthesis. And the purpose of stomates and the leaf epidermis is to minimize water loss from the leaf. But as CO2 diffuses in, water escapes. This simultaneous loss of water and uptake of CO2 means that there is a relationship between the amount of water that's lost and the amount of CO2 that's fixed into dry matter. The transpiration rate depends on things like leaf temperature, wind speed, vapor deficit of the air. And those will affect the amount of dry matter that we get for each kilogram of water we transpire. The range for controlled environment crops is something like 3 to 6 grams of dry matter per kilogram of water. When CO2 is high and the vapor deficit is low, we get something like 6 grams of dry matter per kilogram of water. In ambient CO2 and at larger vapor deficits, typical of greenhouse environments, it's closer to 3. Now, we used this equation in a previous presentation or in this series to compute transpiration. It's the product of a vapor conductance for the leaf and the boundary layer, and the difference between the saturation vapor pressure at leaf temperature and the vapor pressure of the air. So if we knew the wind speed, the stomatal conductance, the canopy temperature, the air vapor pressure, we can compute a transpiration rate. Here's a graph showing transpiration rate versus difference between the saturation vapor pressure at the leaf temperature and the vapor pressure of the air. Now, before we get into it, I want to say a few words about the horizontal axis. It's the vapor pressure deficit of the leaf, or at leaf temperature. Vapor pressures may not be very familiar to you, but relative humidity probably is. A vapor pressure deficit of 0 corresponds to 100% relative humidity in the air. At typical growth room temperatures, a vapor pressure deficit of 2, like maximum extent of the horizontal axis, corresponds to a humidity of about 50%. A vapor pressure deficit of 1 would be about 75% relative humidity. So you can think of the horizontal axis as going from 100% humidity, the left end, and this is referred to the leaf surface, to about 50% on the dry end, on the right end. Now, if we look at the graph and the equation, if the vapor conductance of the leaf were constant, our equation would predict that that blue line would be straight, starting at 0, and it would slope upward. But instead, the blue line goes up and then back down, so that must mean that the vapor conductance isn't constant. The reason is that large vapor pressure differences or low humidities close the stomates. Now, that stands to reason. The guard cells of the stomates lose water too, and it's their turgor pressure that keeps the stomates open. So if they lose water too rapidly, they'll lose turgor and close. We want the stomates to be as wide open as possible to let in as much CO2 as we can get. So we want to maintain the humidity high enough to do that. The exact shape and location of the stomatal response function depends on the species and possibly cultivar. The one shown here is probably typical. The Arroyo Quick Start Guide recommends a vapor pressure deficit of around 1 kilopascal for cannabis. This would be around 75% humidity. The vapor pressure difference shown here is probably a bit larger than the vapor pressure deficit of the air because leaves in light are warmer than the air. But if the response function for cannabis is similar to the one shown here, looks like we would get stomatal closure if we went much beyond the kilopascal, and that's consistent with the information that is in that quick start guide. So we'll come back to calculating dry matter using these two models in a moment, but let's now look for a minute at the nutrient requirements for biomass production. As we've said in earlier lectures, in controlled environments, the substrates typically contain no stored nutrients, so all the nutrients are supplied in the water. Bruce Bugbee, a Utah State University professor, published a wonderful and helpful paper almost 20 years ago, and this table is from his paper. Shows the fraction of the dry biomass made up of each of the nutrients shown. Now, other nutrients are required beyond those shown here, but their mass fraction is so much smaller than these that for our purposes are negligible. The amounts in the leaves, stems, and fruits and roots differ, but if we look just at the leaves, we see that about 10% of the dry mass is the nutrients that we supply. Now let's go through the calculations with each of these models and compare them. Starting with the light, we assume 1,000 micromoles per square meter per second, 12 hours of light. So the daily light integral is 43 moles per square meter per day. We assume 70% of the radiation is intercepted and a light use efficiency of 0.6 grams of dry matter per mole of light. This would give 18 grams per square meter per day. Over 60 days, this would give 1.08 kilograms per square meter. And that's about the value that we mentioned at the start of the lecture. We won't go through the transpiration calculation in detail, but again, if we assume 70% of the light is intercepted, we get 3.2 kilograms of water per square meter per day. We use a dry matter-water ratio of 5 grams of dry matter per kilogram of water. We get 16 grams of dry matter per square meter per day. So that's coming out about the same as it is for light. If we take the 16 grams per square meter per day that we calculated for water and assume that the nutrient requirements are those for leaves, that would take 1.6 grams of nutrients per square meter per day for the dry matter that we're producing. But if you take the 3.2 kilograms of water per square meter per day and multiply by a typical concentration of fertilizer in the irrigation water of 2.5 grams of fertilizer per kilogram of water, now less than half of that mass is the actual nutrients that we need. So we multiply by 0.4 to correct for that. If we do that, we get 3.2 grams of nutrients per square meter per day in the water that was taken up. So now, are these balanced? The light and water seem pretty well balanced. If we were to cut the light to 500 micromoles per square meter per second, that would cut the photons to biomass value to 9 grams per square meter per day. But that change would have very little effect on the transpiration. So those wouldn't be balanced in that case. The nutrients in the water taken up for transpiration appear to be more than can be used for plant growth. But remember, in the second lecture, we talked about using osmotic potential for stressing the crop to do crop steering. Concentrations like those shown here are required to stress the crop. So I think it's clear from that the concentrations of nutrients that we need to do the crop steering are in excess, and will be in excess of those that we need to grow the crop. So what does this all mean? If we know the daily light integral, fractional interception, and light conversion efficiency, we can estimate the light-limited dry matter production. If we know or can compute the transpiration rate, the fractional cover, and the water use efficiency, we can estimate water-limited dry matter production. Now, since the nutrients are supplied with the water, all factors that affect transpiration rate will also affect plant nutrition. Finally, the nutrient levels that are high enough to allow crop steering are likely in excess of those that we would need for the biomass that we're producing. Now, 5 years ago, our company in Pullman was called Decagon, and it combined with UMS, an environmental instrumentation company in Munich, Germany, to form the company we have now called METER. The tagline for UMS was "Measure to Know." That's good advice for controlled environment growers, and here are the sensors needed for those measurements. Starting at the upper left picture, the Arroyo nose has a solar panel that charges its battery, but the solar panel also works as a radiation sensor. Since noses are installed both above and below the canopy, The ratio of the below to above canopy measurement gives an estimate of the fractional light interception. The lower left picture shows meters SC1 leaf porometer clip for measuring stomatal conductance of a leaf. This is a manual measurement. Typical day and night values can be established from these measurements and used for the transpiration calculation. The upper center picture is the Teros 12 water content, electrical conductivity, and temperature sensor for monitoring the plant environment, or the root environment. The bottom center picture is the Arroyo climate station that gives CO2 concentration and accurate air temperature and vapor pressure measurements suitable for the transpiration calculations. The upper right is an apogee quantum sensor connected to the Arroyo system. This provides the light measurements needed for the daily light integral and also for the transpiration calculations. Average wind is probably fairly constant in a growth facility over time, and it could be measured with a handheld anemometer, but if one wanted a record of it, the lower right picture is a meter Atmos 22 sonic anemometer that could be used for continuous monitoring. Now, those measurements are displayed by the Arroyo software to provide the guidance the grower needs to balance the control inputs and monitor resources. We've seen these in previous lectures. This view shows the substrate water content and pore water electrical conductivity for a complete growth cycle. That information is conveniently summarized in dashboards so that you can know the status of all the systems. And here's a kiosk view. And then here's a summary view so you can see the conditions for the whole growth facility. So these are the points that I hope we clarified in today's presentation. Starting with Liebig's law, we tried to make clear that your crop yields are determined by your most limiting resource. We then talked about resource capture by plants in a growth facility in terms of converting photons to biomass, converting water to biomass, and converting nutrients to biomass. The potential conversion rate for photons to biomass is determined by the photosynthetic photon flux density, the CO2 concentration, and the light use efficiency. The potential conversion rate for water to biomass determined by the radiation, the vapor deficit, water use efficiency, and stomatal conductance. Nutrients are supplied by fertigation, and those need to be balanced with each other and with the light and water use, but they're also used for the crop steering. Arroyo tools are designed to help growers maximize production, minimize waste, and to continuously improve. Thank you for being with us today.
All right. Thank you very much, Gaylon. So we'd like to use the next 10 minutes or so to take some questions from the audience. Thank you to those who have submitted your questions already. And there's still plenty of time to submit your questions. So feel free to ask as many as you'd like, and we'll get to as many as we can before we finish here. We do want to let you know that if we do not get to your question, we will either Gaylon or somebody from our meter team will be able to get back to you via email to answer your question directly. All right. So first question here. So is there a way to train plants to collect more light and improve the light use efficiency you were talking about?
I don't know if there's a way to train them to do that. I think there's a way to train growers to do that in a controlled environment facility. If it's understood that photons that aren't captured aren't going to be useful, why there'll be adjustments to the canopy to improve the fraction of radiation that's intercepted. And, a lot of the technique I mean, there are a lot of techniques that are in use right now to do that, to maximize the light interception by the canopy.
Okay. Question number 2 here. Is it practical to try to measure the evaporation in my greenhouse? Can I assume a stomatal conductance, or do I need to measure it? And if so, at how many locations? So combination question there.
Okay, it is practical to compute the transpiration in a greenhouse or in a growth facility, and we've talked about the equations to use to do that and the measurements that are required for it. Stomatal conductance is one of the most important of those measurements, and that needs to be measured quite often. Growth facility or greenhouse, the plants in those environments don't close their stomates at night, and so you need to measure both nighttime and daytime conductance. But if the plant isn't water stressed, why, we can expect the stomatal conductance to stay fairly constant, so it's reasonable to make some measurements by hand and use those for doing those calculations. And how many measurements you need, that's you get an idea as you make some measurements of the variability that exists, and so you can do statistics on that to come up with a reliable number that you can use for the calculation. We'd expect those to change over time as the plant grows and matures, so you need to have some representative of early stages of growth and some representative of later stages.
All right. Okay, do you have any thoughts on how air exchange, airflow, and HPS versus LED affect transpiration rate?
Yeah, the airflow, of course, affects it a lot, especially in controlled environment facilities where the boundary layer conductance is a pretty big fraction of the total conductance for water vapor and heat. And there are equations for that if you know the wind speed, you can know that boundary layer conductance, the wind speed and the leaf size. The radiant energy also has a substantial effect on that, and not just the visible radiation or the radiation that's useful for photosynthesis, but also the thermal radiation, and tend to get quite a bit more heat with the high pressure sodium lamps, and so the transpiration that you get with those lamps tends to be higher than it is with LEDs.
Okay. This next individual is asking, what is the best way to model stomatal conductance and leaf area index? Is there a best way, first of all?
Is there a best way? I wouldn't try to model stomatal conductance. I would just go and measure that with the pyrometer. Mm-hmm. We do a lot of modeling of light interception by plant canopies under outdoor conditions where the plants decide what canopy structure they'll choose, and then that works to model that. But in a controlled environment where the canopy structure is determined mainly by the grower, I'm not sure that that's practical to try to model that. I think probably, again, that's something we just need to measure, but it's easy to measure. So,
okay. All right. Let's see. I think we'll take another question or two here. It looks like you could add more CO2 and light at the same time, and it would help with Liebig's law of the minimum. Are there practical limitations to this approach? More light and more CO2 until what limit?
Well, obviously, we're looking at a pretty small part of the total picture here. Plants certainly have limitations in once the assimilate is produced in the plant, then lots of other stuff has to go on. It has to be transported and stored, those kind of things. And there are limits there that we haven't considered at all here. But you could see from that light response curve that we saw that even at high CO2, we were starting to limit assimilation. And those limitations come elsewhere in the plant. Those are things that we can address with genetics, with looking for cultivars that have increased capability to do that, assimilate, transport, and storage. And I'm sure there's huge variations that exist in that. But those are things that we beyond my ability to explain.
All right. Well, I think that's going to finish it for us here today. We're going to wrap up with that last question there. Again, for those that did not have questions answered, Gaylon or somebody else from our meter group team will be able to get back to you and answer your questions. Thank you again for joining us today. We hope that you enjoyed this discussion as much as we did here. And thank you again for your great questions. Also, please consider answering the short survey that will appear after this webinar is finished, just to let us know what types of webinars you'd like to see in the future. And for more information on what you've seen today, visit us at metergroup.com. Finally, look for the recording of today's presentation in your email and stay tuned for future meter webinars. Thanks again, stay safe, and have a great day.